You may proceed.
Okay. So, hello everyone. So I'm going to present you a part of what I have been doing over the last 2 years, which is the toxicity and biodistribution of 2 lanthanides in Daphne magna after acute and chronic exposure. So thank you, Susanna, because you did a very good introduction of what I'm going to do. I'm not going to really spend a lot of time in telling you what are lanthanides, because I hope everyone here should know what it is. The only thing that I would like to highlight is the fact that because of this high use in modern technology, for example, we are now finding them more and more in the environment, and in my case, my interest in aquatic environment, and that they are now recognized as emerging pollutants, so it's important to measure their toxicity in aquatic ecosystems. So in my case, that's what I'm trying to do in focusing in 2 different lanthanides, which is the lanthanum and the gadolinium. And what I would like to highlight again for this introduction is the fact that lanthanides, they are known to have similar properties. That's what we have been saying this morning. Like, for example, they have similar ionic radius. And some chemists has the tendency to tell us, to ecotoxicologists, that if they have similar properties, they should have comparable toxicity to an organism. So to measure the toxicity, you saw it for people who's not in this field yesterday, that we use some other model organisms. So for example, the Daphnia, which is this small crustacean that you saw yesterday, which is very important in the ecosystem because it represents a very important trophic level for freshwater ecosystem. So to measure the toxicity of lanthanum and gadolinium on this organism, we have different standardized tests that we used, for example, that we present for some of you yesterday, to measure the acute toxicity or the chronic toxicity, like Susanna presented just before. So again, in the case of Daphne magna, the acute toxicity will be a short exposure time, so 48 hours. And in general, we look more at the mortality, where in that case, it's more the mobility. While for chronic toxicity, it's going to be a much longer exposure time, so 21 days in the case of Daphnia. And there we're going to look at more other endpoints, such as the reproduction, the growth, or also others. But today will be mainly the reproduction. So first, to answer that question of what's the toxicity of these 2 elements on Daphne magna, I started with the acute toxicity. And so if you remember, I said that they have similar, I'll say it after, they have similar properties. So in theory, we would expect that it would have similar toxicity to the organism. And so what Susanna also re-explained you is how we measure the toxicity, usually for acute tests. We try to measure this IC50, which is the concentration inhibiting 50% of the mobility. And so if we have different pollutants, we should be able to visualize different type of S-curve, where if the S-curve is a little shorter like this, we will calculate a lower EC50, meaning that the pollutant would be more toxic. And on the other way around, if the EC50 is higher, means that we need a higher dose to get the same effect, so this pollutant would be less toxic. And so if we say that the 2 metals have similar properties, so similar toxicity, we would expect that the IC50 could be pretty comparable. And so that's what I tried first to do, to make this S-curve, to measure the IC50 and to see if IC50 of lanthanum and gadolinium was similar. And so here are the results that I got. And so it's pretty obvious that it's not that easy as what we were expecting. So for gadolinium, I succeeded to get this S-curve. And so the IC50 was calculated approximately at 13.84 milligram per liter. While for lanthanum, it was really complicated. So I measured from 5 to 50 milligram per liter. But after 30 milligram per liter, I had a very big variation among my replicate, which is probably due to very strong precipitation of my lanthanum. So the only thing that I would be sure today to tell you is that the IC50 of lanthanum seems to be higher than 30 milligram per liter. And so then, according to this, I already can say that gadolinium seems to be more toxic than lanthanum, meaning that this first hypothesis is probably wrong. However, it's important to know that this result are according to nominal concentration and not measured concentration, which might also differ according to the result that we have. But so then, my question was, if we have a difference of toxicity, why, like, how can we explain the fact that these two metals with similar properties have a different toxicity? And then my new hypothesis that maybe this difference of toxicity would be due to a difference of toxicokinetics, meaning a difference of biodistribution of way to be uptaken by the organism. So maybe these two metals are accumulating in the organism on a different way. And so to verify this, I decided to focus on one nominal concentration, which was 15 milligram per liter of lanthanum or gadolinium, always separated. And I exposed my organism for 48 hours, and after 48 hours, I took only the alive organism that unfortunately I had to kill to dehydrate in different acetone series to fix them with some HDMS according the technique of Laforge-Tolrien. And the sample, I bring them at Soleil that we visited a year ago to do some X-ray fluorescence measurements at the nanoscopium beam line that you can see just here. And so the idea was to be able to map the distribution of the different elements in my organism. So you can see more precisely here, so my sample was just fixed on a toothstick just here. And here is the beam coming directly to the organism, the two captors detecting the fluorescence, and then the microscope which was connecting to the computer where we're able to scan the zone that we wanted to scan to get this distribution. And so the result that I got are here and are pretty nice, I would say. I was quite happy and surprised to get that. So what is really nice first is that the Daphnia has a carapace made out of calcium, and the calcium is easily detected by this type of technique. And then it was possible to get the complete structure of the organism, which is in that case represented in gray. And then in red, I got the lanthanum distribution here and the gadolinium distribution here. So what we can say is mainly accumulating in the intestine in a quite concentrated way, which is this tube that you can see here, and was also present a little bit in some tissues which was here, and where it's still for me a little bit more complicated to identify what this tissue are, but they might be a part of the thoracic limbs. And then on the other side, we have the gadolinium, where we can see that the metal was much more diffuse in all the different tissue of the organism. And according to different pictures that I have that I cannot show you today, would be a bit too long, I was able to say that the gadolinium seemed to hide in the gills, the body fluids, the maxillary glands, and also a bit in the carapace, meaning that it was more diffuse and in a lot of different tissue of the organism. And so now if you remember, at 15 milligram per liter, this lanthanum did not cause any toxicity on the organism, while gadolinium was causing some lethal effect because it was even higher than the IC50 that we measured. Meaning that according to this, we can kind of validate this new hypothesis that I got, that it seemed that this difference of toxicity is followed by a difference of toxicokinetics. So now if I summarize all of this, we had this first hypothesis saying that similar properties mean similar toxicity, which was refused, because gadolinium tend to be more toxic than lanthanum, and this I also can explain it maybe by the fact that in the test I was working at pH 7, and I saw that lanthanum was precipitating much more than gadolinium. And if this precipitation is happening, there's less metal available to interact with the organism. And if this metal is maybe in particles and in a solid form, this might be easily ingested by the organism, which could explain why this metal is found in the intestinal tract and not on a diffuse form in the organism. And so then from this, I had a sort of new question, which was, as we saw, what was found in the intestinal tract, which might have been ingested, does not seem to cause any toxicity. So then I had my new question, which was maybe lanthanide uptake by ingestion would not cause any effect on the organism. And so to answer that new sort of hypothesis, I decided to do a chronic test that we did actually with Chantal, with another PhD student called Federica, and Edith also helped. And it was a long experiment, so it was good to be together. And so this exposure takes 21 days. And according to the standardized test. This is how people are doing it. So the organism is exposed in 50 milliliter of media, so one organism per 50 milliliter, and they're exposed in spiked media, contaminated media or not. And because it's 21 days, they need to be fed, because for 48 hours you can do the exposure without feeding them. 21 days would be a little bit more problematic. And in the traditional way to do it, we are feeding them with some microalgae that you saw yesterday, but this microalgae are clean microalgae which has been produced in the laboratory. However, in environmental condition, if the Daphnia is growing in a contaminated media, it's going to be fed by an algae which will be also in this contaminated media, meaning that its algae might also be contaminated, meaning that there would be some uptake by ingestion. And so that would be my new question, that if we contaminated this food, do we see more effect or not? And that's some question which have been appearing also on other metals such as copper. So to answer that question, we did this test according the traditional protocol, but in complicating it a little bit more, where we compared 4 different conditions. So the control where the organism were growing in a clean media and fed with a clean food. Then the traditional way to do it, where it's a waterborne exposure, so the organism are growing in a contaminated media, so in our case, 0.5 milligram per liter of lanthanum or gadolinium, but were fed with clean algae. And then 2 new conditions, where we were studying the dietary exposure, so the organism were growing in a clean media, but only fed with contaminated food. So the algae, in that case, were growing in a media contaminated to 0.5 milligram per liter of lanthanum or gadolinium. And then the combined exposure, where they were exposed by waterborne and dietary, so growing in contaminated media and fed with contaminated food. And there, after 21 days, we were looking at mortality, but also at the reproduction for a different type of endpoints, such as the brood. So how many times did they make some babies that we call neonates in the case of Daphnia? How many neonates did we get per brood, when this brood were appearing, and then after 21 days, how many neonates we got in total at the end. So and so if we go back to my hypothesis that the lanthanide uptake by ingestion would have no hazardous effect on the Daphnia magna, according to this hypothesis, we would expect that the toxicity would appear only when the media is contaminated and not when the contamination is appearing via the dietary exposure. So now for the result, first for the mortality, very easy. We did not find any mortality over 21 days when they were exposed to 0.5 milligram per liter. Then the reproduction, it got a little bit more complicated than expected, because according our replicates, we saw that the amount of neonates which were produced by the organism were varying quite a lot, which might be due to a sort of reproduction cycle which is also found in the culture, because even if they are in a control condition in a lab, we saw that the big culture of Daphnia that we have the tendency to sometimes increase in terms of offspring and then reducing also, for example, by the end of the month. And so because of this, we wanted to still be able to compare all this replicate that we had. We always compare the value of the condition according to the respective control, average control that they had, to always compare the replicates according the other. So for the amount of brood, so how many times the organism reproduced, we also did not find any significant difference because all the organisms had approximately 5 broods. However, all the other endpoints were a bit more interesting. So this is what I'm going to try to present now. So first, for the reproduction, let's talk about the first brood date. So at what age the organism started to release their first babies or their first neonates. So we saw that according always to respective control, gadolinium did not seem to affect the reproduction according to this endpoint. However, lanthanum seemed for the combined exposure, so the dietary and waterborne exposure, seemed to delay a little bit this first brood date of approximately 1.13 days. So this was the case for the first brood date. But then we also had the total amount of babies, so total amount of neonates produced after 21 days. And there the results were really interesting because we saw that lanthanum did not seem to affect this endpoint. However, gadolinium were reducing the amount of neonates produced per Daphnia, by 15.8, approximately 15.8 Daphnia in the case of the dietary exposure, and 21.9 juveniles per Daphnia in the case of the combined exposure. And if we go even more in the detail in comparing all the different broods, knowing that there were 5, we saw that it was only the 4th brood which was impacted in the case of the dietary exposure, and it was the 3rd and the 4th brood impacted for the combined exposure, meaning that the 5th brood, which was the last one, was not impacted anymore by this type of exposure. So now, no problem. So now we saw again that according to chronic toxicity, we had 2 metals which were impacting the reproduction, but on a different way. And then I had the same question than the acute toxicity of how is that possible, and does it mean that it's linked to a different type of biodistribution in the organism? So what I did is that I decided to map my elements on the same way as the acute organism, but I have not been to the synchrotron because it takes a bit of time, and my organism were much bigger in that case after 21 days. So I went much closer. We see we are here right now, and this is the laboratory I went, which is the Helmholtz Centrum in Herren, where it's possible to do some X-ray fluorescence measurements with an instrument of Bruker, which is called M4 Tornado. So it does not permit to get such a nice resolution like what we got at the synchrotron, but we still can get pictures and to map the distribution of my metal in the organism. So in that case, the Daphnia were, at the end of the test, were dried in the oven for 48 hours. And you can see they were sticked on a glass plate like this on the side, on double tape, white double tape. And then we were fixing this plate in the tornado, and again selecting the zone that we wanted to scan and get the picture. And so that's the picture that we got. So you see the resolution is not that great in comparison, but it still worked well. So we have the 3 different conditions, dietary exposure, waterborne, and the combined. And then in the case of lanthanum, we see that for the 3 conditions, lanthanum seem to accumulate mainly in the intestinal tract. And the slight difference is that for these 2 conditions, the metal were mainly present at the end of the gut, while for the combined exposure, we were also seeing a little bit on the top, and according to different replicate, it seemed that it's at the beginning of the mouth. Then we had the gadolinium, so it's not really easy to see it properly, but we see that for the dietary exposure, it's accumulating in the intestinal tract that you can see here. It's a little bit cut. For the waterborne exposure, it's much more diffuse, and that's also why it's not that easily visualized here, because the limit of detection is much higher, lower. I mean, it cannot detect such a small concentration of metal, but you can see it a little bit in the organism, which is here. And then we have the combined exposure, where it's also seemed to be quite diffused in the organism. And then in that case, it's a sort of combination between the 2 exposures, because we can find it in the tissue, but also here. So it's not really visible, but we see a small shape like this, which seems to be the intestinal tract, that we were able to validate according different some mapping of other elements, such as the iron, where the iron is mainly accumulated also in the intestinal tract. So we're able to correlate the 2 different maps. So then, if we summarize all of this, if you remember, where we saw an effect on the reproduction, there's 3 conditions: the lanthanum with the combined exposure, and then the gadolinium for the dietary exposure, and the combined exposure. And then my hypothesis that I got before is saying that what accumulates in the intestinal tract will not cause any effect. It seemed that it's not that easy to validate that hypothesis according to chronic exposure, because we can see that here, the metal accumulates in the intestinal tract, but it's affecting in delaying the reproduction, while here the dietary exposure also accumulates the gadolinium in the intestinal tract, and we saw that the reproduction were reduced. So all of this is kind of creating a lot of new questions that I now have, because, for example, how to explain that the lanthanum, regardless of the condition that I had, were accumulating on the same way. And this might be due to precipitation again. Maybe lanthanum is precipitating even at 0.5 milligram per liter, explaining why they could be ingested by the organism. And we're still waiting the soluble measurements by ICP-MS, so I cannot really tell you more about that. But another possibility could be that even if we give some clean algae in the media, if the lanthanum has a very strong affinity for the algae, all the lanthanum would go in the algae, and then the lanthanum which will be uptaken by the organism would be also by ingestion. So this is only hypothesis, and I cannot really give you more information about this for now. And then there's another question, which is, does it mean that there's a sort of adaptation by the organism, knowing that in that case, it's only the first brood which was impacted, but not the other. And here, the brood which were reduced in terms of babies per Daphnia were only the third one, and here the fourth one, and here the third and fourth one. But the fifth brood were, again, normal compared to the control. So there's possibly a sort of adaptation that the organism are developing over the 21 days, and that could be interesting to also expose them for longer time exposure to validate that possibility or not. So now if I summarize you all my thought regarding this presentation, so I had lanthanum gadolinium could have comparable toxicity, and according to my result, it means that no, and this could be explained by a difference of toxicokinetics, because as you can see, lanthanum accumulating this in the tract is not toxic, causing toxicity, while gadolinium is much more diffuse in the tissue and was causing some toxicity. And so then I got this new question that uptake by ingestion is not toxic, and so with this chronic exposure, it seemed that it's not that easy to conclude like this, because we saw that even dietary exposure is affecting the reproduction. And so there we have a lot of new questions, but at least the conclusion that I could have is that the combined exposure for lanthanum and gadolinium were affecting the reproduction, even if they are affected on a different way, meaning that dietary exposure has to be taken in account for chronic toxicity, which is different to the standardized protocol which is offered by the different association. So thank you for your attention.
Thank you very much, Marion. I don't know if maybe we have some questions in the crowd. Easy.
Yes?
Hi, very nice presentation.
Thanks.
I have a question about the last slide on the results, where you say that the dietary conditions give you the toxicity also in the reproduction. Have you considered that maybe the fact that you are feeding contaminated algae at the end increases the level of the rare earth element that the Daphnia gets? I mean, because if you contaminate algae and then you put the contaminated algae at the beaker where the Daphnia is growing, you really can't control how much is the Daphnia eating. So at the end, maybe the level of the rare earth elements is higher from the other condition. I don't know if that makes sense.
Yeah, so for the...
So maybe it's, I mean, maybe the higher effect is affecting the reproduction change in this case.
Yeah, so we wanted to measure the internal concentration in the organism. The problem is the amount of biomass, and we never have enough. So that's a big problem that we kind of had. And unfortunately, in that case, with the measurement that we did with the Bruker instrument, it was not possible to measure, to quantify somehow the difference of intensity between the different exposure. So that's a big question that we also have. However, also regarding the food of maybe if the food is contaminated, they are eating less or more. That's one endpoint that we also studied, because we studied also the growth. I mean, we studied a lot of different endpoints. And according to the results, all the organisms seem to have the same type of feeding behavior. They seem to eat the same amount of cells, that they are contaminated or not.
Okay, but I mean, or at least I think it this way, that if the algae is contaminated, and you have a principal beginning concentration that you said 0.5, then at the end, you have the
you mean
chain,
the transfer?
No? So at the end, the food chain. So at the end of the Daphnia, probably it's more than the algae. So this is what I meant.
Yeah. And that was kind of the question of this dietary exposure. Is it causing something or not? And I think it was, I mean, still expected, according to what Chantal is doing, that this food chain is actually increasing the uptake and then might also affect, in terms of toxicity, the toxicity of these elements.
That's a great job.
Yes.
Yeah, first of all, congratulations for the pictures in
Yeah.
the synchrotron because we were not that lucky.
Yeah, I was there and I remember.
And the second question is my vision, my perspective from hydrogeology and reaction. I see that the effects you have mentioned is 15 milligrams per liter is really high.
It's a lot. Yes.
So,
Yeah.
I'm not aware of a river or in an industrial effluent with
No.
such
That's
a
for
high
sure.
amount. Is this, the question is this frequent in your experience to find that high concentration, or is it because of the species, or is because otherwise these are very good news if the toxicity is found at this high concentration of lanthanum in this case, or this is because it's a species that is quite resistant?
I think the Daphnia is pretty resistant still, according what Zafia is doing for some other pollutant, that it seems that it's a species which is particularly resistant. However, the IC50, which are measured for most of the organism, are still pretty high. So yeah, it's a sort of good news. But again, it's an acute toxicity, and that's why it's really important to work in chronic toxicity, because we get at much lower concentration, and there we might see more information on it.
I'm telling this because I think from the Panorama point of view of the project, and this is something that is not done normally in the ITNs or in the projects, I think it's good to start thinking about messages. Messages rise from the project.
Yeah.
And this could be one of the message from the project telling, well, our contribution in general as a project is that some species are really resistant to rare earth toxicity or something like that. It's just to start, because at the end the project ends and everyone is dispersed, so no final message from the project is sent.
Well, however, I'm not sure that I will use this conclusion,
No,
because no problem, we don't care. No, I think because these tests are standardized tests and they are made in really simple condition. And then that's where, for example, what Chantal is doing in checking more this food web model and understanding on a longer time exposure and understanding this accumulation by ingestion. And there we can work at much lower concentration and we still can see some effect. And so that's why, yes, according to the standardized test, there's not so much problem, but this standardized test allow other researchers to then work on more representative condition to the environment.
What also must be kept in mind is that this is one specific species of Daphnia magna, and Marion and I also performed an algae test where we look at different species of algae.
Yeah.
And we used algae species that's not recommended by the ISO standard. And we saw that this species is actually way more sensitive than the species that is recommended by the standard. So there are a lot of factors and variations that play into a role, and it's very dangerous to already kind of make this conclusion.
No, what I meant is that when
But I agree, yeah, for conclusion.
we write the proposal of ITN, we have a general objective. And everybody knows that each PhD project has a very specific details which are just written or shown in a scientific journals. But in terms of messages to general public or scientific community in general, we wrote an objective in the proposal, and then we should write the conclusions.
Yes, this is totally
right?
true. Yeah.
I'm talking about the general project, okay? And now I think it's interesting to start thinking about these general messages, okay?
Yeah.
I understand, and this is different from what we publish in the journals.
Yeah,
we
sure,
go
sure.
to the specific species and so on, but I think it should be interesting to start thinking about the general message. Because we wrote something in the proposal as objective.
Yeah.
You said that you calculate your EC50 using nominal concentration. Did you try to use measure
Yeah.
concentration?
So I have, I think later on I had some other nice picture. Yes, here I have the measured concentration that I got according lanthanum and gadolinium. And so today I was only able to talk with nominal concentration because I actually cannot measure anything with lanthanum because the soluble concentration even decrease. So that is for sure that even talking about difference of toxicity between lanthanum and gadolinium is complicated to say even just with this result because we cannot measure an IC50 and maybe the IC50 according to measured concentration would be similar between lanthanum and gadolinium because if we see for gadolinium the IC50 is between 10 and 15, so between 6 and 7 for soluble concentration, and lanthanum we never succeed to get more than 2.9 milligram per liter. So that is for sure there's a big difference, and that is with acute toxicity I did not succeed to measure anything, and that is why with EDIT we did some tests with 7 days of exposure, and then we succeeded to measure some difference value, but I think she's gonna present them after it. Yeah.
I have a question for a non-scientific
Uh-huh.
point of view. Could you explain to me what is the difference between nominal concentration?
Yes. Nominal concentration is the concentration that we put in the media, that we prepare. So it's what we wanted to do. And then you're going to have some complexation as the metal will react with different elements and can also precipitate. So at the end of the test, we measure the soluble concentration that we have at the end. And as you can see, there's a big difference because I was planning to work at 15 milligram per liter, and lanthanum I was at 2, and gadolinium at 7.
Yeah.
But that is also because I was working at pH 7, and as Antonio said, if we decrease the pH, we have a much higher toxicity because there's more elements which are soluble during the exposure, during the test.
This is measured before the test?
At the end.
At the end of the test. But you still consider the uptake of the organism, right?
There is also some, but I need to look again at, because I also measured some at the beginning.
Okay, and they still change from the
I mean, actually, no, I think it was not that different. So there's for sure that some metals are also taken by the organism, but there's also the fact, and I think that's also, we have been talking about it with Mukit, that the speciation modeling, I think, is sometimes complicated to do in ecotoxicology, because what we can model is what happened in media without an organism. But if we add an organism, this organism might release also something, and the complexation might differ, and we might also have more precipitation. And that's what we did, with microalgae experiment. We measured at the end of the test with and without algae, and we have concentration which are completely different. Yeah. Yeah.
When was the end of the test after
48 hours.
48 hours?
Yeah.
And why did you measure the real concentration at the end of the test?
Sorry?
Why did you measure the real concentration at the end?
I think the idea is to know what is available at the end. And I think, to be honest, that's something that I will need to look more in detail, because for me, I understood that this is where we can get approximately the equilibrium and get really an idea of what is actually really bioavailable for the organism.
Did you measure the real concentration also in the beginning?
So for this test, I need to look. I remember that we did it with 15 milligram per liter, and in the case of gadolinium, we were not so far from 15. But
Okay, so
for the Daphnia, I really should do it again. But with the algae, at least I'm not sure that we did it. I remember that we did it really at the beginning, and we kind of forgot. And that I agree that it would be something really interesting to look at.
But you also did this kind of time series, didn't you? To look at.
Yeah, but it was not all the concentration, and it was quite some time. I mean, I think it's some time ago, and I don't have the exact results, so I prefer not saying anything about it.
In our lab, usually when we calculate the real concentrations, we do it at the beginning of the experiment. So to know, for example, we prepare the sample of 15 milligrams per liter. We want to see exactly at that time if it is indeed 15 or we did a mistake. So
And
I think it's more important in the beginning than
In the beginning?
And at the end, we measure the organism and the water, so you can understand how much did the organism get
Okay.
and how much is left in the
Exactly,
sample.
yeah.
So the IC50 that you're making with measured concentration or from beginning of the test?
It's in the beginning, exactly.
Yeah, then that might be interesting that I do it too. Okay.
We use this concentration.
Important is also to get this time series really
Yeah,
talked about.
yeah.
And I think we should have some.
We have some with Edith, but I don't
data
remember
on that.
which concentration this was. But yeah, we did some, but it
Because
was
you have
also
all
a few
these
months ago.
absorption,
Mhm.
It was for,
as
I think
well.
up to 50, was it that's too high, no?
No, not 50, because it was with the 7-day test, so
No,
we
it
would
was-
need to look. I mean, yeah.
Question or do you want to have
Coffee is good.
Thank you very much for
Yeah, I don't know what I did.
I do
Not
have
sure
a
this is-
question, but I was afraid to ask it in front of